Hello again, everybody, and welcome to the Scientific Origins channel, where we bravely and curiously explore the universe to find if the data we collect matches our proposals and scientific models attempting to explain the universe. Today we're going to talk about another common evolution denial trope. This is one you'll encounter frequently. It's one that evolution deniers use to try and pretend like, oh, we're just skeptical. We're just not convinced because the evidence isn't strong enough. Show me where evolution has been observed. Nobody has ever seen a creature become another creature, yada, yada, yada. So, we're going to address why this argument is wrong today and point out many instances where evolution has absolutely been observed. And some of these are my favorites, and hopefully you'll pick out one or two that you like and that you can go look up and learn more about, and you can counter evolution deniers when they spew this bullcrap. So, the reality is that it takes a ton of planning, foresight, uh, understanding of what evolution is and how the mechanisms of it work to actually observe a speciation. It takes uh, just nothing short of absolute genius to predict that a species is going to evolve over the course of a certain period of time, and then to figure out how to prove it, how to show it, how to record the observations necessary that shows that a speciation event has occurred here. This is macroevolution. Yet scientists understand evolution well enough to do exactly that. We're going to go over a couple examples here. So, how have we observed evolution and speciation? And you can see here, unfortunately, my uh, face is blocking the last one, but that last one just says cetaceans, which we're actually not going to cover in this video. We're going to do another video on cetaceans. Cetaceans deserve their own video entirely, so we're going to do a video specifically dedicated to the set Artiodactyla dataset, that is whales, orcas, and dolphins. But you can see these other examples here. We've got the marbled crayfish, greenish warbler, cichlid fishes, finch hybridization, Baltic Sea algae, Italian wall lizards, Nordic mollusks, niche partitioning, London mosquito, Drosophila, coprophagous beetles, genus Solanum, Teosinte, choanoflagellates, brassica, and cetaceans. I put 15 examples that prove this argument wrong. Of course, you could go all day just listing rows after rows. I put these three because these 3 rows of 5 because these are some of my favorites, some of the ones that I know well enough to talk about them. So we're gonna just go down the list by order here, Jeopardy style. So, marbled crayfish. Marbled crayfish. This is one of the clearest examples of speciation ever observed. So, when we talk about a species, typically what we are referring to is what's called the biological species concept. And as it turns out, the biological species concept is broken. It's arbitrary. It doesn't actually work. There are species that are considered different species from one another that are actually capable of producing fertile hybrids or fertile non-hybrid offspring. For instance, a wolf and a poodle are technically considered different species, but they can successfully produce offspring together, and their offspring will be fertile. I think in most cases, when you hybridize a wolf with a non-wolf canine, what you're going to get is something that looks almost completely like a wolf. It looks like a wolf non-hybrid. It is technically a hybrid, but it looks like a non-hybrid. So this is what the species concept refers to. It refers to species that are capable of producing fertile non-hybrid offspring together typically. And the further away a species is from able to be, two organisms are away from being able to do that, the further away from being the same species they are. And in the case of the marbled crayfish, we had a single mutation event that produced a new crayfish that reproduces asexually. It's parthenogenetic. It does parthenogenesis. It has a different number of chromosomes, different genome regulation, and is reproductively isolated from its parent species. It is a new species by every way that science tries to define what a species is. This thing changed its number of chromosomes. It's reproductively isolated from its parent species. In fact, it doesn't reproduce with any other species. It's parthenogenetic, and it has a different number of chromosomes and different genome regulation. It's It's a very strong proof of evolution, and it proves that evolution can generate new species with new genetic architecture, not just minor variation. The greenish warbler. So the greenish warbler is a great example of what's called a ring species. And I could have put ring species specifically in here, but I think we're going to end up doing a video about ring species specifically themselves because there are many examples of them. Greenish warbler is one, and Satina scoltzi is another. There's lots of ring species. And what a ring species is, a ring species is when you have a founding population, and then the founding population and a subset of that population get separated, usually by geographic isolation, and eventually the subpopulation changes, but it's still interfertile with the original population. And then the subsequent population, the subset of population A that became population B, a subset of that gets further isolated and becomes population C. And now population B and population C are interfertile with each other, but population A and population C are not. So even though population A and population C share common ancestors, they're no longer reproductively compatible, and that is what a ring species is. And this is a classic example of it. With the greenish warbler, populations spread around the Tibetan Plateau, gradually changing as they expanded. Neighboring populations interbreed, but when the two ends meet again, they can no longer interbreed. This shows speciation happening gradually with no sharp boundaries, exactly as evolution predicts. So species break down into a continuum. Cichlid fishes. These ones are fun because it's not only a great example of evolution, they're also just really pretty to look at. Look at all these colorful kaleidoscopic arrays of cichlid fishes. And how we got all these different arrays of cichlid fishes is that they rapidly diversified into hundreds of species with different jaws, diets, behaviors, and mating signals, all from common ancestors. So these changes are genetically mapped and tied to ecological niches, and it demonstrates how adaptive radiation can produce many new species with new structures in geologically short time spans. So each of these different colors, different shapes, different mandibles, they allow these cichlid subspecies to thrive in a more unique niche within the niche that cichlid fishes fill. And as a result, they've evolved rapidly into many, many different varieties. So the cactus finch hybridization. This one is fascinating, not just because we observed a new species of finch emerging from the hybridization of two previously existing lineages. These are the finches. They're the ones. These are the finches that Darwin himself observed, and it's a further demonstration of how the scientific motto of "we stand on the shoulders of giants," because we're standing on the shoulders of Darwin, but we're also standing on the shoulders of two other giants here who were themselves standing on the shoulders of Darwin. It's giants all the way down, as you were. And those two giants are Dr. Peter and Rosemary Grant, a couple who lives on the Galapagos. They're a married couple. They love bird watching. They love science and ornithology and evolution. And they've been watching over the last 30 years as this hybridization event occurred and a new species of cactus finch emerged. I believe it's called Big Bird. Baltic Sea algae. So algae, phytology, I think, is what you call the study of algae, and it's an incredible tool for evolution and biology because algae evolves very rapidly. We are capable of observing species of algae evolving in all kinds of different ways. So due to an environmental change, these algae became reproductively isolated from its ancestor despite living in close proximity, and this shows that speciation can occur without geographic isolation. It can be driven just by ecological pressure and genetics. Other relevant studies of algae have shown things like algae evolving to be multicellular and different cells within the new multicellular colony of algae specializing and becoming more capable of doing specific tasks within the cellular colony. The force that drives multicellular evolution is cooperation, and we'll talk more about that a little bit on another one of these examples. So the Italian wall lizards, these things in just a few decades, these lizards that were transplanted, they were brought along on migrant boats they transplanted this population and it evolved new gut structures, changes in skull shape, bite force, and behavior. It allowed it to shift from insectivory to herbivory. These things were carnivorous. They went from subsisting on a diet of nothing but other living organisms. Well, plants are living organisms too. Nothing but other animals to subsisting on a diet of pure plants. So they evolved herbivory, and it was a fascinating discovery because these lizards got transplanted. The settlers that transplanted them were like, when they left the island, were like, we're just going to leave them here. They're carnivorous, so eventually, and they're invasive, so eventually they'll eat all of the things that they can eat, and then they'll die out of starvation. We don't need to exterminate them. And then humans returned to the island a long time later, I think decades later, and they discovered that these lizards were still there, still thriving. And what had happened was they had evolved. They had altered their niche from an insectivorous niche to an herbivorous niche, and they're now fully herbivorous, which involves some huge evolutions that had to happen in order for their diet to completely change. This is one of my favorites. This one's really cool. So this one shows how understanding evolution lets you do clever, clever things. So the clever, clever thing that happened here is that these species of mollusks, when we start our story, there's two species of them. There's a below tide and an above tide species. And this is an example of what's called niche partitioning. So when niche partitioning occurs, you have two species that are filling a similar niche, but they're not quite exactly the same. In this case, the niches are very similar, and the difference is that one species is filling this niche above tide, and the other species is filling it below tide. And then pollution happened. The nutrient levels in lakes were off the charts. Fertilizer runoff is a real problem in agriculture. It's something that pollutes the environment by over nutrientizing lakes, and these lakes became way too high in nutrient levels, which caused these choking mats of algae to form, which as a result, this pollution caused the above tide species to go extinct. Their they weren't able to get the food they needed to survive anymore, while the below tide species still was. So the below tide species still exists in this polluted state of the lake, and then laws get passed. The pollution gets outlawed, and scientists make a prediction. Because the two species exist previously, they predict that as the pollution is outlawed and the lake returns to normal, there will be a selective pressure for something to fill that above tide niche again, because the nutrients are there once more. And then they watched over 30 years as their prediction came true. There's now once again two mollusk species there, an above tide and a below tide species, and you absolutely should check this study out because it's one of the coolest things I've ever read about. It took about 60 generations over about 30 years, and a speciation occurred, and the second niche was once again filled due to the mechanics of niche partitioning. The London underground mosquito. So this one you've probably heard about before. It's talked about a lot. It's a classic example of geographic isolation. You have an above ground mosquito species and an underground mosquito species. The underground mosquito species doesn't have as much access to the above ground mosquito's main food source, blood. So it evolves a new survival strategy. It stops being a hemovore and it starts feeding on other food sources. And as a result, you produce sexually selective prezygotic barriers, a speciation. They're now different species. The Diane-Dodd incipient sexual separation of Drosophila melanogaster. I probably need to just make a video about this one itself too, because what a cool experiment. So what happened is Diane-Dodd, she took two populations, as this graph shows. She took one population at the start at the top there. You have that one Drosophila population, and she separated it into two different populations, geographically isolated them, and she changed the food source in the population B. Population A kept subsisting on the normal starch diet, and population B was subjected to a maltose diet. And then over about 40 generations, took about a year, you ended up with incipient sexual separation, meaning the population that was subjected to the maltose food had undergone enough genotypic and phenotypic changes that the descendants of the original population would no longer select them as mates. They wouldn't select each other as mates at all, and they were unlikely to select each other as mates, even in scenarios where their mate selection was artificially restrained, where they didn't have any choices but this other population. They still recognized each other as a different species. And one of the things that defines what a species is its behavior, what it will try and mate and replicate itself with. So another great example of obvious speciation. The coprophagous Scarabidae mandible diversification. This one is super cool. Not only did we produce a speciation, we also produced a novel feature. We took a population of beetles that didn't have a mandible, and they evolved a mandible by messing with the beetle's environment and creating selection pressures that select for first mandible-like protrusions that made them better to manipulate things. The more those protrusions developed, you eventually ended up with a full-on mandible. So not only is it an example of speciation, it's an example of how selection and mutation can create novel features by just tinkering with what's already there. Selective breeding. So this one is really obvious. This one is something everybody should know about. If you take a dog with stubby little legs and you breed it with another dog with stubby little legs, you get the Dachshund, right? And that's what selective breeding is. It's humans replacing the selective force of nature, and instead the selective pressures that get exerted on the population are shaped by human desires. So ethnobotanical evolution, ethnozoological evolution, that refers to species that humans, our ecological relationship has caused a lot of their evolution. So we see some examples here in this picture. We see fruits and vegetables that humans have shaped, and that has been very useful for our survival as we've adapted and developed. So we're going to talk about a couple of those. There's first the genus Solanum, which is the genus that includes tomatoes, potatoes, and the nightshade plant, eggplants. Many of these species are all from the genus Solanum, and they've been created by humans doing artificial selection on them. You might not know this. This is an interesting fact. Because the eggplant, the tomato, the potato, all of these are descendants of the Solanum genus. Their leaves are toxic. So you can eat a tomato plant, you can eat an eggplant's fruiting body, you can eat the parts of the plant that have been shaped for human ingestion, but the leaves are still as toxic as nightshade ever was. So there's some clues about their ancestry, and it's a fascinating balance how we've been able to take something that was toxic and turn it, part of it, into a food source without changing the toxic part of the plant at all. Then there's Teosinte. This is another great example. I'm probably going to end up doing a whole video about Teosinte itself as well as probably brassica. But Teosinte is an example of evolution created by selective breeding. Humans took this wild population of seedy, mildly edible grass called Teosinte, and they selected for genes controlling the size of the kernel, the softness of the kernel, the sweetness and aroma and color of the kernel. And by selecting these genes, they didn't know they were selecting genes. They were just selecting the traits that were favorable to them. By selecting these genes, they ended up modifying this species of wild seedy grass that looks just like any other grass into corn, which most people would not be able to tell as grass just by looking at it. So, yeah, and another important thing about the Teosinte, creationists will say, okay, yeah, I see all this evolution, but you can't create novel features, and we kind of address that with the Scarabidae, but you can address it with the Teosinte as well, in that, you know what Teosinte doesn't have? A cob. But corn has a cob, and all a cob is a modified stem. New feature created by modifying old features, evolution. And then brassica. This is the last one on the list here. You might not know this, but kohlrabi, broccoli, kale, cauliflower, like 100 different species are actually all just descendants of the brassica, the wildflower, uh, that we have evolved into all these different forms of produce, which we do by selecting the genes that control for size of different parts of the plant. So, like for instance, Brussels sprouts is created by selecting the genes that control for size of leaf buds. Kale is created by selecting the genes that control for leaf size. Broccoli is created by selecting the stem, and so on and so forth. So that's brassica, another great example of evolution, and a great example of how evolution improves our lives by giving us something to eat. Choanoflagellates. Now, these ones are really interesting, and I think it's kind of funny because this is a picture of Volvox. I started to get a little bit lazy here at the end of it. These aren't choanoflagellates. These are Volvox. These are, Volvoxes are an algae species where the individual Volvox exist inside of a membrane and cooperate. It's an intermediary stage between unicellular and multicellular evolution. So the image works, but it's not a choanoflagellate. If you want to see a choanoflagellate, just go on Google and look up a choanoflagellate. So what choanoflagellates are is they're little swimmy boys. They're little microscopic species, and these are actually what sponges, sea sponges, poriferans, are made out of. They're made out of these choanoflagellates, which are, I think they're considered apoicozoans, whereas choanoflagellates are holozoans, or not choanoflagellates, poriferans are holozoans or metazoans. It's like one step. We're talking about very, very early stages of evolution here, the evolution of multicellularity. So choanoflagellates are precursors to sponges, and we know that because if you get enough of them together and you watch them, they will evolve into a sponge in observable time spans. And then interestingly, if you put a divider in, like a grid-shaped divider, they'll evolve back into being unicellular organisms. You can introduce a physical barrier that directs their evolution back towards unicellular organisms. So they can evolve from unicellular to multicellular and from multicellular to unicellular, which shatters so many creationist arguments. So there's an extremely high number of these species. Poriferans, there's thousands of them. They're one of the largest phylum in the animal kingdom, along with echinoderms, annelids, planarians, chordats, that's us, and so on and so forth. But poriferans are one of the largest ones. And these things exist without even having organs. They don't have organs, they don't have tissues. They're just a colony of individual cells that have learned to cooperate so they can exist as a sessile filter feeder with a hydrostatic skeleton that just pumps water through its system and filters the nutrients out. And they've done this very well. So because they exist without even organs or tissues, they completely destroy the argument of irreducible complexity. You can see how even before organs or tissues exist in a system, how just a loose colony of cells can evolve into a multicellular organism. And they're pivotal to our understanding of the evolution of multicellularity, which is based on cooperation and specialization. So, why do creationists say this? Why do evolution deniers say this? Why do they say this so obviously wrong thing that I can disprove with dozens of examples right off the top of my head? And why have these examples not stopped them from saying these things? So first of all, you can't discount ignorance. Probably most creationists, evolution deniers, they don't know what a choanoflagellate is. They don't know what a cichlid fish is. They've never heard of a greenish warbler. These are all just mystical things that don't exist to them. They don't know about them. So is the first problem. The second thing you're going to encounter is going to be straw manning and lots of hand waving. They're going to say, oh, a fruit fly evolved into a fly. Wow, what an impressive evolution. Or they're going to say, you didn't show a species becoming a completely different species. I want to see an amoeba evolve into a whale. So they're going to straw man and they're going to hand wave. But what this actually shows, this shows reproductive isolation. It shows that one population can become two populations even though they share common ancestors that are completely reproductively incompatible with another. This means that you can get different populations. You can have one population become many populations, and you can get disparate adaptations stacked up on top of their fundamental similarity, which is exactly what we see when we look at organisms. We see the mammal clade, and it's separated in all of these different kinds of mammals, carnivorans and artiodactyls and so on and so forth. But they all have the chordat, vertebrate, metazoan. They all have those characteristics that come before they diversified into their own specific group, and then they have those unique characteristics that put them in that group stacked up on top of those basal characteristics that group them into the larger group. So there's no hard, and it's important to also notice that when you show them this, they're going to say, well, that's not good enough. But these are just single steps. These are single steps. This is a speciation happening, and you're going from one population to multiple populations. There's nothing that stops each of those populations from becoming multiple populations and from compounding change occurring until you get all of biodiversity. And the burden of proof is on them to show that there is some hard barrier that stops that speciation from accumulating such that evolution cannot account for all biodiversity, and no such thing exists. So that's it. That's it for this one. I hope you guys had fun. I hope you learned just how full of it evolution deniers are when they say that evolution isn't observable. I hope you find these examples fascinating and look into some of them yourself. And as always, thank you for coming to the Scientific Origins channel, where we bravely and curiously explore the universe and try to find if the data we collect matches our models and propositions for how the universe works. We're proposing quantifiable, falsifiable solutions to unsolved problems and unanswered questions in natural history, and using what we learn to better human existence. And so that is why science is important, and why science helps us all, and why anti-science bullcrap shall not be tolerated and must be pushed back against. Thank you very much.